I'm Bill Newman. I'm a clinical geneticist in Manchester, for those of you who haven't met me. And what I'd like to do is just to give you a little bit of a background as to what the GMSA, the Genomic Medicine Service Alliance is, and where it fits into sort of the genetics, genomics sort of architecture across the country, and then give you a little bit of an update as to some of the things that we've been doing over the last couple of years and what's coming next, if that's okay. So the whole concept of the genomics medicine service sort of emerged around the time of the 100,000 Genomes Project, sort of 2012, when David Cameron was sort of running that. The important thing to say is that clinical genetics and genetics laboratories have been around for a very long time. And the 100,000 Genomes Project and being able to deliver genetic testing in a way that hadn't been delivered in other countries by using whole genome sequencing was only possible because genetic services had worked so effectively for a large number of years. So I always find it sort of a little bit frustrating when people seem to think that genomics was invented in 2015. Because that absolutely isn't the truth of it. But we have seen an enormous investment in genetics and genomics, both in terms of the technology and in terms of some of the opportunities that this branch of medicine allows. That said, it's important to recognize that there are challenges, there are things that haven't gone as well as we want to. And one of the roles of the Genomic Medicine Service Alliance is to try to look at how we can ensure that genetics is delivered for our patients and their families as effectively as possible, recognizing that not everything works as smooth as you want it to straight off. So in 2018, as the 100,000 Genomes Project was coming to an end, and that was a project run in England, delivering 100,000 genomes in individuals with rare conditions and with cancer. And there's probably been about a third of the people with a rare condition who have got a diagnosis through that. So it's important to recognize there are still 2/3 of the people who took part in that program who don't have an answer. So there's an ongoing piece of work that needs to be done there. And then there is this consideration whether whole genome sequencing would be a useful test in patients with cancer to get them onto the right treatment. I think the jury's still out on that. I think there are lots of things that we can do in patients with cancer to look at their tumor to get them onto the right treatment. I'm not sure if whole genome sequencing is always the answer. And it's really important to remember that just because we have a test or a technology, we need to think about how do we use it and how do we use it most effectively, and don't allow ourselves to be purely driven by the technology and some of those interests. So in 2018, there was a paper published by NHS England, which set out a vision that there would be a new laboratory service, such that there would be 7 laboratories, major laboratory hubs around the country. So one in the northwest, so that is based at Manchester, but with a large laboratory here at Liverpool Women's Hospital, and with also connections to Liverpool Clinical Laboratories and the Christie Hospital, where there's testing happening. So it's a network of labs across the northwest, and that's mirrored in other parts of the country. The best thing about the new laboratory service is that all the genetic testing now is nationally commissioned. That means that if you're an oncologist or a cardiologist or a dermatologist and you want to deliver a genetic test, you don't need to worry about who's going to pay for it. Because if your patient meets the criteria which are published, then you can get that test. So that's a fantastic thing, and means that it's far more equitable. Because I know that in the past, it was very much, you know, could you get genetic testing dependent on where you live, that postcode lottery. So that's gone. However, there are some challenges in delivering the testing at the scale that's required, and that's an ongoing piece of work. Although it must be said that the laboratory in the Northwest, which Emma Howard runs, so Emma used to run the lab at Liverpool Women's, and now runs the whole laboratory infrastructure, has done a remarkable job with her team. And the waiting times for getting tests in the Northwest are shorter than anywhere else in the country. So that's the laboratory side of things. We then also have clinical services. So there's a clinical genetic service here in Liverpool at Liverpool Women's. We provide services across this region. And then there's one in Manchester as well. And we provide peripheral clinics across the Northwest. And over the next few weeks, the new service specification for how clinical genetic services should be delivered will be published. So that's been under draft for a number of months, but it will be published sometime in June. And then that hopefully the funding will come that will allow those services to be delivered. But like lots of clinical services, there are real challenges because we have staff retiring, we don't have enough trainees coming through. Trying to ensure that there's this handover of some of the things that clinical geneticists would have done in the past to other specialties is still challenging. Outside of the clinical genetic service and the laboratory service is this Genomic Medicine Service Alliance. And I think of it a little bit like the cancer alliances. So we don't have a patient-facing role. We don't see patients as such. We're not responsible for their care. But what our remit is to do a few things. So one, to complete some of the work around the 100,000 Genomes Project. So there's ongoing work about giving further results out from that. It's working with colleagues in different specialties. So not just doctors, but nurses, midwives, pharmacists, different health professionals. Because if we are going to make sure that genetics is relevant for all the patients who need it, we need all the health professionals that are interacting with those patients to know what they need to know about genetics and how it's useful. So if you're working in accident and emergency or in orthopedics, you probably don't need to know much genetics. But if you're a pediatric nurse working in neurology, you need to know lots of genetics because a lot of the conditions that you're seeing those children with are inherited, and it's very useful to be able to provide those families with the right information. It's also to work around improving the access to genetics and the provision of that, and I'll touch on that in a second, and then a number of projects that we've been running, which I'll talk through. So we've linked in with a number of people across the Northwest to make sure that what we're doing isn't focused just on a few people in Manchester, but stretches across the whole region. So linked into a number of organisations like the Cancer Alliances, public health physicians, the chief nurse, chief midwife, and of course patient representatives at the core of what we're doing. And we've been looking at how we can improve people's understanding of genomics, both in the community and other health professionals. And so on our website, you see that we've got an equality strategy. And one of the projects that we've been doing is working very closely with the Pakistani and Bangladeshi communities, because there's been very little involvement for that community in terms of some of the accessing some of the benefits of genetic medicine. So we see in Manchester, as I'm sure you do, a large number of families of Pakistani origin where we can offer a genetic test to see the cause of the health problem in the family, maybe a child with severe learning disability or congenital birth defects. But often when the genetic result comes back, it's what's called a variant of uncertain significance. We can't really interpret it. We're not really sure what it means. And one of the large reasons for that is because when you identify a genetic variant, one of the things you want to know is how common is that variant in the general population? Is it seen lots of times before? Because if it's seen quite regularly, it's not going to be the cause of a very rare condition. But if you've never seen it before, then you can be more confident that it is really the cause of that condition. But if you don't have any data from particular populations across the world, then you don't know, is it more common in that population or not? So you can't interpret it. And so the South Asian population makes up sort of 5% of the population across the UK, but in certain parts of the Northwest, it's much higher than that, 20%, 30% in central Burnley and Blackburn. And if you look at the disease burden in that population, Pakistani men have a 3 to 4 times higher risk of cardiovascular disease, of type 2 diabetes, and there was a really severe burden of disease associated with COVID over the last 3 years. And if you look at the UK Biobank, so UK Biobank was this study of 500,000 people aged 45 and over that took place about 20 years ago, and we're seeing now some of the benefits of some of the research from that. If you look at the people who participated, only 3,000 of the 500,000 were either Bangladeshi or Pakistani in origin. And so the amount of data we have from that population is tiny. And so we need to increase that. And so that's why we're working with colleagues in Bradford and East London to identify people who wish to take part in a study where we are accessing samples and clinical data so that we can understand some of the genetic reasons for some of these common conditions, but also provide a data set which we can compare in our routine clinical practice. So one of the things that I mentioned was that there were a number of national projects set up. And so each of the 7 regions around the country took a lead on various projects. So in the Northwest, the project that we took a lead on was led by Munir Pir Mohammed, Liverpool, who's a world expert on pharmacogenetics. And it was about introducing this new test called DPYD into clinical practice. So patients, particularly with breast cancer and bowel cancer, who were getting a drug called 5-FU or capecitabine, we knew that there was a risk of toxicity associated with that. 1 in 1,000 people would have an absence of this particular enzyme made by this gene. And if you've got an absence of this enzyme, it means you're at very high risk of really severe toxicity, even leading to fatality. We knew that by introducing testing that we could reduce not all, but a lot of that toxicity. So one of the things that we were keen to do is if you introduce a test, to make sure that everyone's using it, that the test results come back on time, that then the doctors and the pharmacists are using the information from that testing to change practice. It's just not another test that we think, oh, I don't need to use that. We've never had that in the past. That they're actually using the results meaningfully. And so that's the project that we led in the Northwest and worked very well. One of the things that we noted, though, was that at the moment, we only check for 4 variants in that gene. And if you come from other parts of the world, you're not white European ancestry, then there are probably other genetic variants that are relevant that we need to look at. And so we're continuing to look in detail at what other variants we might need to test to make that testing more inclusive. So there are projects around maturity onset diabetes of the young. So Sabnam Samad here in Liverpool led that program. So that's a very rare type of inherited diabetes, probably affects about 1% of all diabetes. The reason it's important is because you don't need insulin, you need tablet medication, or sometimes you might not need any medication. And at the start of the project, we knew there were probably about 700 people in the Northwest with that condition, and we hadn't reached all of them. And so Sabnam set up a network with all the endocrinology and diabetes units across the region to make sure that there's an awareness of that the testing is free and people are being identified with that. I won't go through all the other projects, but just to mention the Sudden Cardiac Death Project at the bottom, because that's ongoing. We've known for a long time that if a young person dies suddenly in unexplained circumstances, there could be lots of reasons for that. One of them could be that they have an inherited heart muscle problem, a cardiomyopathy, or they have a heart rhythm problem. Over the last number of years, there's been genetic testing that can help to look at that. So you can take a sample at post-mortem, do genetic testing on that sample, and see if you can find the explanation for that person's death. When a family is referred to us, I think that we do that fairly well. The family are seen by cardiology team, either at Broad Green or Alder Hey or in Manchester, and the genetic testing is all organised. One of the problems was, however, is that a lot of the families weren't being referred to us. There wasn't a mechanism to do that. So that programme which Kim Clark, who's one of the genetic counsellors in Liverpool, has been leading the Northwest, has allowed the coroner's officer to directly refer into us, so then we can organise to see the family, make sure that the right samples are taken, the right consents are taken, the heart, if it needs to be examined, is seen by an expert pathologist, and then the family is all seen in a timely way. So that project's now open in Liverpool, and we're just about to open it up in Blackburn. And the plan is by the end of the year to have it running as a standard service across the whole of the Northwest. So over the 2 years of the project, we've had a number of small projects which have been regionally based. So this isn't happening in the rest of the country. These are projects that we've led on because we think we've got some local expertise in the Northwest that we think if we looked at these in a bit more detail, then we take some learning from that and we share that more broadly. So you can see there's some projects around pharmacogenetics. Vicky Mackay's been doing a project in Liverpool, looking at some patients with inherited heart conditions. You might want that result really quickly if you think it's going to direct whether that patient needs surgery or they might need a device like an implantable defibrillator, and that decision is going to be made based on whether they've got a genetic change or not. We worked with the alkaptonuria team here in Liverpool as well, because they run a national service and they were keen to try and work out how they could link in with the genetic services around the country to provide the local support that their families required, rather than them always coming to Liverpool. We've got a number of projects this year where we've worked with social workers at the needs of looked after children who have been adopted, who might not know their biological family, but when they're thinking about having a family of their own, might want to sort of explore are there genetic implications for them, or if they have health problems, what testing should be undertaken in those. We've been looking at whether we should provide genomic counselling services for patients with mental health disorders. And we've been looking a lot with Rosie Lord here at Clatterbridge across the region, working with Cancer Alliances as to how the right samples are taken from patients with cancer. They're then sent to the laboratory for the right test, and that information can be fed back appropriately. So I'm just going to give you an example of a piece of work that we've been involved with that's been close to my heart and hopefully sort of illustrates some of the things that we're trying to do. So about 30 years, well, it's not about, it is 30 years ago, a group in America and Israel found that there was a change in a gene that if you were given the antibiotic gentamicin, that you are at very high risk of going deaf with that gene, due to that gene change. And it's found across the world, about 1 in 500 people. And so if you're a child with cystic fibrosis or a bladder problem, you're almost certainly going to get given gentamicin at some point in your life. It might not be tomorrow, it might not be next week, but at some point in your life, you're going to get that antibiotic. And so you can do a test and you get the result back a few days later, and then depending on that test, you can see whether gentamicin is going to be safe or not in that context. But we were aware that there were a number of children, well, newborn babies particularly, going onto special care baby units where the decision about giving them gentamicin needed to happen within the first hour of them being admitted to NICU. So if you've got a test where you take a blood sample, send it to the lab, you get the result 3 or 4 days later, that's absolutely hopeless in the context of needing to treat that baby there and then. And so we looked to see whether we could develop a test that could give a result at that point, so at the bedside. And so what we did was we developed a test with a small spin-out company in Manchester where you take a cheek swab, and then every baby coming into the unit, so this happened at Liverpool Women's and in Manchester through the COVID period. And what we saw that the testing got a result back in 26 minutes, so that's within your hour. It gave you time then to prescribe the drugs, and we saw that there was no difference between when the testing didn't happen at all and when the testing was introduced in how long the baby got their antibiotics. During the study period, we we tested 750 babies, and of those, 3 of them had the genetic variant, and they were given a different antibiotic, so didn't go on to become deaf. And so you can avoid preventable hearing loss in these children. So that was an interesting and a good outcome, but it was a study and it was research. So what we wanted to do was see, can we roll that out into routine clinical practice? Because that's where, you know, there's this valley of death. It's a great research project, but it doesn't actually happen in clinical practice. And that's what we're trying to do in the GMSA. We're trying to take good ideas and see if we can use them in clinical practice. So one of the first questions, it's a new test. It's very complicated. It's fancy machinery. It's going to cost a load of money. Well, it does have a cost, but if you offset that against the cost of those children, 200 every year in the UK, who would have the genetic variant, not going deaf, they don't need to have bilateral cochlear implants. And then there are all the added benefits of them not needing additional support because of their hearing loss. So if you do the health economics, you probably save somewhere between £5 and £7 million a year. And NICE have just looked at this and have made an early value assessment. So they're going to give approval for this to be rolled out across the country. And what we're doing now is we're working with some units across the Northwest so that it's not just in the big NICUs that this happens, it's in small little units, in small hospitals across the region that we can show that we can do this, and that then hopefully that this will be introduced as a routine clinical intervention. So that's all I wanted to say. It was just an example of where I think we can make a difference. I'm happy to answer any questions if you've got any at all. Stanley, yes.
Very impressive. I mean, it's somebody who's reaching retirement, I'm going to miss out on all this, but Rob Johnson, public cardiologist here. I'd just like to get some feel for just the economics. Not because we shouldn't spend the money, but just I presume these tests, these sort of whole genomic testing is coming down and down in cost. Would you be able to give us a feel for like how much it's got cheaper in the last 20 years?
Yeah. So I mean, it's truly remarkable. I mean, I don't think I had the imagination to think that I would ever be able to order a whole genome as a clinical test. So I remember vividly Bill Clinton and Tony Blair on the White House lawn talking about the whole human genome and how it had been sequenced. That cost $3 billion for one genome and took 15 years to do. Now, the costs people say are probably just for the reagents, and you could get a genome done for somewhere between $100 and $200. And that can be done in about 2 weeks or so. It costs more than that in the NHS because you've got the analysis, you've got to pay for the staff, you know, but still the scale of how cost effective it is remarkable. There are challenges with it though. There's still a lot of most of the things that we find, we don't understand what it means. And so we can overinterpret things, we can make people very anxious, we can give people information that they don't know what to do with. And also, as we've always had in medicine, you know, there is this challenge around incidental findings. So if you do a chest x-ray on someone, you can find out that they've got lung cancer. If you do a full blood count, you know, for somebody, you find out they've got leukemia. That's always been something in medicine. It's something that happens and can happen in genetics, and we need to be very careful about how we manage that. And also, not only does that have implications for the individual, but of course for their children, their siblings, and all that sort of stuff. So I'm very wary of doing whole genome sequencing or genetic testing in people who aren't unwell. If you have a particular genetic condition or a condition and genetic testing can tell you something useful about that, I think that's absolutely fine. But I've never had a genetic test. I've got no intention of having a genetic test until I need one. And I could have had one if I wanted one. There are quite a few geneticists that sort of think it's interesting and there's a bit of vanity genetics going on. You know, I know what's his name, Ozzy Osbourne, and all these people having their genomes done, and isn't it all a great laugh? Well, it's a great laugh until you find something that you don't want to know. And you shouldn't have genetic testing until you've spoken to those closest to you, your partner, your children, other people in your family, because it's not all about yourself when it comes to genetics, it's about you as a group. And so I love genetics. I think it's remarkable what it can do, and I'm very passionate about it, but we absolutely mustn't just allow ourselves just to be driven by the technology, because frankly, it's getting to a time where all of us in a rich country can have our genomes done. That's not the challenge. The challenge is, what do we do with it and how do we use that information? Any other?
Well, actually, nobody else got.
Yeah, follow-ups, yeah.
There's all this unparalleled sort of, where does AI fit into this? Because this data is presumably very AI-able.
Yes. Yeah. No, so genetics is absolutely ripe for AI in a number of different ways. I think the big areas where AI is going to have a big impact over the next few years are going to be in radiology, pathology, where you've got images and you just train the programs to look at those images, and they just get better and better at it. People in our laboratories, the clinical scientists, are super highly trained scientists. I mean, they're really smart people. They spend lots of their time looking at a genetic variant, sitting in front of their computer screen and trying to work out, is that variant the cause of that particular person's condition? It takes them days and weeks to do that. That's where the delay is in getting your genetic test results. It's in their meticulous way of going over that data. The way they go through it is by, they need to look up on that database, they need to look at that database, they need to look at that database, they need to double check it. All of that will be done by software, by AI, quicker and more accurately, with less variability between those people. And so we've been trialing AI in our laboratory. The initial predictions are that it's going to reduce the amount of work by about 80%. It's going to completely transform how quickly you get back your genetic results over the next few years. So you think, oh gosh, well, that's great on one hand. What's going to happen to all those scientists? There are loads and loads of things that they can be doing in the laboratory, loads of other tests that help to interpret what those genetic tests mean. So they will all have jobs, they will all be doing more interesting things than sitting in front of a computer screen. So I think it's going to be a remarkable benefit over the next little bit. We've got to be careful with how we use it, because if a mistake gets into the system with AI, it just gets replicated and that mistake gets bigger. So you always need people going in and checking and making sure that it's okay. But it's going to be a enormous shift and positive shift, I think, for us. Yes, hi.
Towns Information Agency, the AHSN. I'm just interested in what the other next year's projects will be. So year one.
Yeah. So that's that's a great question, and I didn't touch on it. So the NHS England unit, who coordinates all this, have decided to change their model. So we're not going to have regional projects this coming year, which I think is a terrible shame, because I think we've learnt a lot from doing the projects we have, and I think that we know better in the Northwest what projects that will be useful to our population. But that's not my decision. What they are doing is setting up what's called networks of excellence. So these are going to be a number of initiatives. We're at the moment just waiting for the outcomes. We've just got a feedback from our expression of interest, and we should know in the next few weeks. We've put forward a programme of work working with Munir, particularly around pharmacogenetics, because we think that's a real strength in the Northwest, which probably isn't mirrored anywhere else in the UK. So we will be working with other colleagues across the country, but I think the real strength is between Liverpool and Manchester. We will be involved in a number of the other networks of excellence, not necessarily leading them, but involved in them. We don't know the outcome yet, but we believe that there'll be projects focused on cancer, on rare conditions and how you diagnose those using different techniques. There's some talk of a project around AI and how that can be looked at and used more effectively. But once we know the outcomes of those, we'll put it all on our website, we'll try and disseminate it, and we'll try and make sure that people have an opportunity to say, oh, well, we've got some strength there, we've got some knowledge, we'd like to link in with that network so we can be a partner in it. Great, okay, I'm free. Thank you.
Think. Right, okay, so moving on. I think the slight change in emphasis so from diagnostic point of view, looking at the treatment side of things. So I'm one of the neuromuscular consultants here. So let's start with the case. So this is a little baby who was born on time, born in good condition, and was found to be a bit weak, floppy by about 3 to 4 weeks of age, and was diagnosed with a condition called spinal muscular atrophy type 1. So this is a condition where you get progressively weak and floppy infant who are thought to be cognitively well, kind of very alert, and then they progressively get weaker and essentially don't survive. So as you can see, it is a condition where you progressively lose your motor neurons, and then if you don't have your nerves working, then your muscles don't work, it gets weaker, and babies need support with feeding, breathing, etc., and but eventually they all die within the first year, within the first 18 months of life. So if I'd seen this baby a few years ago, maybe 3 years ago, the only thing we could offer was a kind of symptomatic, care, palliative care, and that was it. So they would have gone on and die. The reason for that is you lose your motor neurons at a very rapid pace in the first few weeks and months of life. So we think of it as an emergency at this point. So when I see these patients today, I can offer a gene therapy. So I can fast track them to get gene therapy in a week or so. So we are very keen to see these babies as soon as possible, diagnose them, and start treatment. So I've got 2 videos. I'll show you the first one, hope it works. So this is a baby who had the gene therapy with SMA type 1, who had gene therapy at 8 weeks of age. So he was diagnosed around that time and had the treatment. And you can see how this baby is doing. So this baby is around 10 months of age now. What?
I'm Charlie, I'm 34 and we live in St Albans. This is Rupert. He is a little over 10 months old now, and he was diagnosed with spinal muscular atrophy type 1 when he was 8 weeks old. As you can see, Rupert loves his farm animals, and you're going to speak for yourself. And he just loves generally playing. He's very chatty.
So just to pause there. So as you can see, he's alive. He's not on needing any ventilator support, not on any feeding support. But he is still not normal. He's weak. As you can see, if you look at this video, he's not moving that. He's got very little.
Started to read books with us.
He's not doing what he
loves just closing the book, don't you? So we've got lots of pop-up books, and he's forever just tugging at them. And I think it's just any way of sort of getting him to move. It's almost intentional physio, which is terrible to say, but we do a lot of playtime that's making.
So yeah, so that's it. So yes, so we can make their life better. So we can make them survive, but we are not quite there yet. So we're not, improving their life dramatically, but not to the extent that we would like to, change. But if you treat these patients before they get any symptoms, so this second video is somebody who had treatment before they develop any symptoms. And so for that, we need newborn screening. And so we can see
for her missing SMN gene.
Babies with SMA are missing a gene vital for development. Through a one-time injection, gene therapy replaces this missing gene. For Evelyn, this gene therapy worked.
We start seeing changes as early as 2 months after treatment.
She started to push, and she started to get more active, holding her head up. And a little after 3-4 months, she rolled over on her own.
Now 3-year-old Evelyn challenges Dr. Mendel to a dance-off at her annual follow-up appointment.
She comes back after 3 years and she runs up to me and hugs me and says, "Dr. Mendel, I love you."
So that's pre-symptomatic patients. So this, if you treat them much earlier before they get any symptoms, they do much better. So it's been borne out of all the studies that we know can support this idea. Right. Okay. So moving on. So SMA is one of more than 500 single gene disorders that we see in neuromuscular group. And many of these other conditions are going the same way as SMA, where there is some kind of treatment trial going on, which is very likely to come to clinical use in the near future. So I think we have to say the recent advances in genomic medicine as a whole had a positive impact, both in terms of diagnosis as well as in treatment. So when we see patients now in our clinic, the first test we do is a genomic testing. So we used to do muscle biopsies, MRIs, this, that, and the other. So we don't do any of those now. So we go for genetic tests. It could be a single gene test. It could be a whole exome, whole genome, whatever it is. And I think we've been one of the first centers in the UK to do this. So we started doing a genomic testing as a first-line test soon after the 100,000 Genome Project, and we had various labs offering gene panel testings, which we used to do, and we had fairly high diagnostic rate. So for example, in a limb-girdle muscular dystrophies, we had about 80% diagnostic rate, which is quite good. So that meant that we were not routinely using the various other investigations, muscle biopsies, etc., for our patients. So it was kind of cost saving, less expensive, less invasive. We don't need a GA for this in our patients. That was all really good. And we now use this kind of testing more as a functional test. So if you see a variant of unknown significance, then we use maybe muscle biopsies to kind of confirm whether we are seeing something there or not. So it kind of the emphasis or the reason for doing this test has changed. And hopefully we'll have other tools like transcriptomics or proteomics coming to us in the near future. So who knows? So what has this led to? So this has given a fast diagnosis. So genomic testing as a first-line diagnostic test means we get a fast diagnosis, more precise diagnosis in many cases. We are seeing new phenotypes. So if you look at any genetic condition, if you look at one gene, it gives you a spectrum of problems. Not all of them are described in the textbooks. So we see things that are not in the textbooks. So between whatever, the milder to the more severe. So all of these phenotypes, there is a spectrum we see. We are seeing better, we are seeing newer diseases, newer disease pathways. So I spend a lot of time reading, basics now, cell biology and, all of those things. I'll show you some examples of that. It has led to improved management. Sometimes it's palliative care, so we can deal with or kind of manage it much earlier than before. And for most of the clinical trials now and for treatments, you need a genetic diagnosis. So that kind of drives or kind of directs people to the right trial and treatment. So these are some of the genetic diagnoses we had in the last few years. And so if you look at the pathways that these conditions involve, so we never thought of a muscle condition as a trafficking disorder or a disorder of autophagy. So that is what we're dealing with. So that means you have to spend a lot of time reading around this. What is, how is this controlled? And, it's not what we study in our medical school. Well, certainly not me, or in our last, 10, 15 years. And if you look at, so this is a recent publication, it's the International Classification of Inherited Metabolic Disorders. So it describes a lot of these conditions. So if you look at, there are more than 1,500 new disorders being described in the last 4 or 5 years, all driven by genomic testing, because there are no biomarkers for these conditions, there are no, typical phenotype. Well, mostly don't. They are multi-system disorders. So it's not an easy diagnosis to make clinically. And hopefully there is a newborn genomes program being rolled out, hopefully like 100,000 genomes program, and that might give us more early diagnosis in many of these cases. So shifting on to the treatment. So when we think of a genetic disorder, we think of something wrong with the protein functions, whether it's a structural protein, whether it's a contractile protein, signaling protein, channels, whatever it is, hormones, enzymes, etc., etc. And most of it is driven by something wrong with your genetic coding. Sometimes it can be even before that in terms of chromatin, remodeling. Sometimes it can be at the RNA level, or etc., etc. So when we think of treatments broadly, we can think of those that target the gene, called gene editing, different ways of it. Those that target the RNA, called RNA technologies. And from a neuromuscular point of view, it is mostly pre-mRNA splicing, if you want to be specific around it. And then more commonly, the gene therapy or gene addition therapy, we call it, or gene replacement therapy. And if you look at all the trials that's happening now, most of it is either gene therapy or RNA technologies. Gene editing is at a very early stage, and it is in preclinical or early clinical phase. So very quickly for gene addition therapy or gene therapy, so the idea is that if you have a faulty gene, you give a new gene to the patient, and you use a vector to deliver that gene into your cell. And once it reaches the cell, the gene then gets into the nucleus, start producing the protein, and then the condition improves because you've got the functioning protein now. So most commonly used vector is AAV, or adeno-associated virus. So the wild type virus is modified to put a gene into the virus. The advantage of that is it is a non-pathogenic virus. So it doesn't cause any human disease. It's very good at infecting all the cells, both dividing and non-dividing cells. And once it gets into the cell, it stays there for a long time. Long time meaning, at this point, we know it certainly stays there for at least 10 to 15 years. So we haven't got any data beyond that. The major other advantage is it does not integrate with the host cell genome. It stays as an episome. It kind of stays within the nucleus, and a circular DNA. It doesn't go and attach itself to the host genome, which means there's no risk of off-target effect or mutations in the genome. So challenges and disadvantages, it is a very small virus. So the capacity is low. So we can't put large genes like dystrophin, for example, or dysferlin. So it need to be either broken down to 2 parts or have a micro dystrophin or a mini dystrophin type changes. It is immunogenic. So any virus will cause immune response from your body, either to the capsid, the viral cover, or to the gene itself. In terms of muscle conditions, it does not transfer the stem cells, which is a problem. So over time, you can get a dilutional effect. And there are concerns about how long this will stay in the nucleus, whether it needs redosing after 18 years, 20 years, who knows. So there are a number of gene therapy, either trials or treatments available now. So Zolgensma, as we saw, is the first one. And the second one has just had a positive recommendation from the FDA's subcommittee, and there will be a final decision made in the next few weeks. So this is the first gene therapy for Duchenne muscular dystrophy. So DMD, this is a Pfizer gene therapy. We we are doing a trial here. We dosed our first patient a few weeks ago for this Pfizer trial. But it's all not, rosy. I think what we know is for every condition, it behaves differently. So we cannot use one solution to treat all the genetic conditions. For example, then this is an X-linked myotubular myopathy, where the gene therapy was stopped because there were kind of 4 or 5 deaths out of about 10 patients because of liver complications. And this has never been seen in animal models. So we had a lot of data from animal trials which showed no problem with the liver, but as soon as you gave it to the patients, start having liver complications. Right. So what are the other technologies? So RNA technology is very common. So we use a number of these medications in trial now. So ASOs or antisense oligonucleotides, I won't go into detail because of time issues, but they're very specific. So it goes and attaches into a very specific region of your genome. So there's no off-target effect mostly. But the chemistries aren't very good. So I think the existing chemistries are not very good at going to the right tissue at high enough doses or levels, and it's got limited bio-oral availability. So you can't give it oral medication. So you need to be intravenous or intrathecal medication because it cannot even penetrate the blood-brain barrier either. Costly. All of these medicines are very costly. Another attempt is small molecules. Again, it does work at the mRNA level, alter the splicing. And this time it can be given orally. So it's got good oral bioavailability. So we have patients on this medication. So you need to have it every day. The problem here is it can bind to many targets. It's not very specific for your SMA, for example, in Risdiplam. So there's been a recent publication where cell studies show that it can affect up to 1,000 gene function. So function around 1,000 genes, and around 200 to 250 genes, you can alter the function by at least 20%. So I think we don't know the long-term effects of this drugs. Gene editing, I won't go into detail, but there are different ways of gene editing. CRISPR-Cas9 is a bacterial system of essentially, if a virus invades bacteria take a piece of virus and puts it into its own genome to protect against further viral infections. So that is a system that can be used to cut and paste or to do gene editing. And there are newer techniques. So these are in very early clinical trials. Right. So coming back to SMA, so there are 3 different types of SMA, mainly in pediatric population. And it is because we haven't got an SMN1 gene, which is the main gene that produces most of the SMN protein. But there is also a backup SMN2 gene, which produces about 10 to 15% of the good protein. Now, 85% is not a good protein, but about 10, 15%. So the idea of some of these mRNA splicing therapy is to convert SMN2 to an SMN1 gene to produce more of that functional protein. So we have 3 medications for SMA at the moment. So we have about 25 more patients on one of these treatments. So the first one is an antisense oligonucleotide. It's an intrathecal injection given 4 monthly after the initial loading phase. The second one is a small molecule, which is a daily oral treatment. Third one is a one-off intravenous infusion. And you can see the cost. So they're not cheap. So the gene therapy is $2 million for the one injection. That's the list price. I think NHS gets it at a slightly lower cost. Yeah, so lots of questions around cost, which I'll come on to in a minute. Just going back to the SMA newborn screening. So for the gene therapy or any of this treatment to work well, you need patients to be diagnosed at birth or even before birth. Because we know that for SMA with 2 copies of SMN2, there are changes even by second trimester. You see an abnormal neuromuscular junction by second trimester. So this condition starts even before birth. So in the US, 99% of all newborns are screened for SMA. In Europe, things are improving. A number of countries are doing kind of starting screening now. And in the UK, there is a pilot phase going on, and hopefully we'll have newborn screening in the next year or two. I think if you look at the rest of the world, the situation isn't that great. Most of the countries in low middle income countries don't have either treatment or screening. So that is a problem. And again, I won't go into detail, but there are, you do a two-step approach to screening these patients, and it is very cost effective. It is got high sensitivity and specificity, so it's a very good test. Some countries have moved on to carrier screening. So this is you're screening the parents or mom and dad. So some countries like Israel, for example, they have a lot of experience in doing carrier screening, and they have a kind of close to eliminating SMA in their population, like many other conditions like Tay-Sachs and things. There are a number of advantages for this, but I think it does not detect de novo mutation, which can happen in around 2%. And newborn screening, we said, is very cost effective. So we just briefly go through various drug pipelines. So SMA, the first 3 medications which are targeting motor neurons are already in the market. We use it. The next 3 or 4 are targeting muscle because it is a muscular, it also has a muscle problem. And mostly these are myostatin inhibitors. And then a number of trials beyond that, which are in either preclinical or early phase clinical trials. And I can look at all other conditions that we see, and all of them have got some kind of trials going on. The next one is Duchenne muscular dystrophy. A lot of exon skipping trials. We use the Translarna or atelurin for nonsense mutations. But a number of other trials, medications have been approved by FDA, not by EMA or MHRA yet, because they think it is not cost effective. Again, in gene therapy, this is only a matter of time before these medications come to use. We can look at various other conditions. So limb-girdle muscular dystrophies are most common for 5. I've got a gene therapy trial. FKRP is the last one. So this one, which had the first patient dosed a few months ago. Pompe's, Friedreich's. So Friedreich's ataxia had the first medication approved by FDA in March. So this is a NRF2 activator. So it's a transcription activator. Myotonic dystrophy. So I can look at, take a list, neuropathy. All of these conditions have lots of trials. And we do a lot of research here. At any one point, we have about 6 or 7 trials running. So we, as you can see from that list, a number of them are exon skipping trials. There is a gene therapy trial, as we said before. We completed the Givinostat trial, which is a HDAC inhibitor. It kind of alters the chromatin remodeling. And there is a myostatin trial, which is coming up. And there are about 7 or 8 trials which are, going here in the next few months, years. So benefits, it's good. It gives hope to families with rare disease where there's a high unmet need. It's mostly highly effective, at least in the short term. Improves survival, better quality of life, fewer comorbidities. In some cases, it's a single dose treatment. So you may not need another dose at all. Challenges, there is always risk around immune response. So if you got anti-AAV antibodies, you are not able to get treatment. So about 5 to 10% of children around the age of 3 will have antibodies. If you're living in certain countries, so anti-AAV is very common in monkeys. So if you live in one of the Asian countries or where lots of monkeys, so there's close interaction, you've got much higher AAV antibodies. So we may not be able to treat many of those patients. But then you can also get immune response against your virus, against your antigen that you're giving, and to the gene that you're giving. Second challenge is managing expectations. So people think this is a cure. It isn't a cure at this point. It is, you're modifying from a severe to a milder condition. So that is the best we can think of. So it is, yeah, so it's a lot of work around managing expectations, especially around, when you do take consent. For gene therapy, we're giving them in a few days to make up their mind. Do you need a gene therapy or not? Yes or no? The parents are under pressure to make a decision within a day, within a few days' time. And it isn't easy for them. So we have to support these families with psychology, counseling. So a lot of work that goes around getting consent for these treatments. There is uncertainty around long-term efficacy and safety, as we discussed, and the high cost of drugs. It is a problem. How do we afford it? How do we give access to people around the world? And ethical challenges. Do we treat one patient with a $2 million drug, or do we treat 10,000 patients with a much lower cost-effective intervention? So I won't go into details, but yeah, so a lot of them are costly drugs. So across the world, there are many interventions, many kind of initiatives to try and find an answer. So some are country specific. So this is a French collaboration to try and find a cost-effective genetic treatment. Genathon is one of the non-profit company who's had the first gene product for DMD, which is undergoing trial now. Another one is, so there's a Knowledge Ecology International. This is a non-profit organization in Washington. They were very successful in getting treatment to low and middle income countries, for example, in HIV, and they're now taking up risdiplam as the next medication that they want to give to add to the essential medicines list. There's also there's something called a Bespoke Gene Therapy Consortium in the US. It is a consortium of government, industry, and non-profit organization. Again, with an aim to find cheaper gene therapy, genetic treatment alternatives. And China is coming, so they are very good at technology-wise, and the expectation is that the cost will come down as China starts producing more and more of this, and there will be more competition. Okay, right. In summary, so we had very positive impact on neuromuscular treatment and diagnosis from the recent genomic advances. A number of challenges remain: resource constraints, side effects, concerns about long-term safety, efficacy, etc. But overall, I think we are at a much better place now compared to even 3 or 4 years ago in terms of what we can offer and the hope that to children and families. I'm not sure what the time is, but I'm okay to stop. Right. Any questions around?
Take one more question. How, if you suspect that a baby has SMA, how long would it take from the bedside to get a genetic result back, a positive or negative?
It depends on where you live and which lab it goes to. Yeah, even in the UK. So if you are in Edinburgh or in Scotland, I think they get the result within 72 hours, if I'm not mistaken. So here we get it, if you ask the lab. So I think partly so if you just send the blood off to the lab and say SMS, test for SMA, which many people do, then it goes to the lab, it sits there, it doesn't get tested, so it might take a few weeks. If you ring the lab and say, I've got a patient with suspected SMA, I need a testing urgently, we can get a result within a week or so. In the US, you give the sample in the morning, you get the result by evening, and they treat so they give genetic treatment by day 2 of life, day 3. So that's what it is doable. If we can be done in the US, there's no reason why we can't do it here. We need people in all of those things to work. Any other questions? Thank you. Shall I close? Okay, yeah.
No. No.
Hi everybody, so my name is Catherine Breen. I'm another consultant geneticist from Manchester. I'm also the Deputy Clinical Director of the North West GMSA. This is a project that's one of the regional projects that we've been doing. We're sort of in the process of finishing it off now, but we thought to give you a quick flavour of it because I know we're sort of heading towards our break. So just to say that most of the work has been done by my colleague, Michelle Bottomley, who's a genetic counsellor in Manchester, and I'm sort of presenting on our behalf today. The other thing just to mention is that I'll sort of say looked after child, but it means care experienced person. So that can be somebody who's a child who's currently looked after or has been at some point in the past or has perhaps been adopted. So the aim of the project was to improve access to genetic services for care experienced people who've got a personal or family history of an inherited genetic condition. So when information perhaps has come to light or somebody's been diagnosed with an inherited genetic condition, and it's based in the Northwest. So the first challenge was to understand what's actually going on at the moment, and then try and find some areas for change, and then agree those areas with the key stakeholders. So what are the problems with delayed access to genetic advice and testing? And I think it's quite timely and following on from Dr. Madhu, who's been talking about sort of that delay in diagnosis and what that can mean potentially for treatment. But there are other conditions where if there's a delay in treatment, it can be a lot of uncertainty for families, and that can lead to this problem called the diagnostic odyssey, where people can feel like they're on a long journey to get to answers. And there's also, as people get older, there's potentially, if they don't know they've got a family history of a genetic condition or don't know they've got a genetic condition, there can be some missed opportunities for them themselves to miss out on screening, for example, or to modify life choices. And also, again, with following on from Dr. Madhu, with some neuromuscular conditions, for example, there can be people who are carriers of a genetic disorder, don't know, it doesn't affect their own personal health, but it can affect sometimes when they make decisions about reproductive choices. So this is a pathway that we've worked out through doing this project about what actually happens at the moment and the different sort of points at which genetic information can become available. So there should be an initial health assessment within 28 days of becoming a looked after child, and there are these sort of standardised forms that are filled in. Then a child might go on to a pre-adoption medical or might go on to have regular health assessments as part of being a looked after child. Now, at any point in that, there could be testing for a genetic condition that's presenting with symptoms. Or as part of the health assessment, information might become available perhaps from those parental forms about the risk of a late onset condition or risk of future reproductive risk when somebody's older. And then at any point in this journey, there can be ad hoc information that comes in perhaps from the biological family, and it can come via clinicians, social work, carers, foster carers. And the initial study idea was very much to think about this ad hoc information that comes in and how that can be linked up perhaps with a child who's not in contact with their biological family anymore. But as with any good study, hopefully, the initial study idea was the starting point, but as it's developed, we've sort of realised that it's more complicated and thought about different areas that we can potentially have an impact as well. So sometimes it can become difficult. So these are just the questions we started asking ourselves. Sometimes it can become difficult to verify information that's provided, particularly when it's come through quite a convoluted route. There's also sort of some inconsistency in how often sort of updates to family history information is being sought for looked after children. And again, we thought about whether there could be guidance around that. Once there's information available about, for example, a later onset condition in a child who's looked after, are the sort of professionals involved with that child, do they know what information to then pass on? And could that mean that there could be better links with genetics to get that sort of advice? When there's testing that needs to happen for a symptomatic genetic condition, are the genetic appointments quick enough? Are patients waiting a long time when it might have an impact on, for example, their plans for future care? And the other thing we started thinking about was we're increasingly asking, for example, social workers to give consent for tests like whole genome sequencing. And are they well equipped to do that at the moment, or could some extra training be helpful? And thinking about the opportunities for gathering extra family history information and where they could come in. Forms that are standardised, are they consistently used? They're sort of, they should be, but are they being used? And if they aren't completed at the time, for example, when a child becomes looked after, are there other opportunities for gathering the same information? So what have we done so far? So we've had lots of meetings and several focus groups. So we've had contact with Adoption Now, who work across the Northwest, and we've had a focus group with their post-adoption social workers about the challenges that they face and asking for some feedback about how we might be able to help. We've also attended the Northwest Looked After Child Specialist Nurse Forum and again completed a focus group. And we've had a meeting with a social worker who's doing a project at the moment about improving healthcare for children who are looked after. And she's asked us to be involved to sort of give some input around genetics and genomics. We've also had a meeting with the University of Manchester about including some genomics in the social work undergraduate programme as well. And we've met a care experienced representative who works for NHS England at the moment as a patient expert, and she's going to be reviewing any materials that we produce, including any training. And we've developed multiple questionnaires to gather further information and adapted one for community paediatricians as well, because obviously they're integral to this process. So again, I'll just whiz through these, but we're thinking of developing some training, and that would be for social workers, for genetics, for community paediatrics, depending on the outcome of the questionnaires. And a sort of idea that we've come up with across this project is about thinking about a key contact within a genetics department for people who've got questions about looked after children, about these sorts of things. It can be quite complicated, depending on the condition, depending on the context. So having one person that, for example, social workers know they can contact with these questions might be helpful. And again, sort of reciprocating that, a key contact in social services departments who has a little more knowledge and training around this might be helpful as well. Another idea was around sort of when there's an annual health review, making more of it being more of a standardised process that family history information is asked about again, just to see if new information has come to light. And then also at the point where somebody volunteers information about, say, a new diagnosis in the biological family, at that point when the contact is made, could a consent form be sent to ask for access to records to be able to verify what that condition is and think about the implications for the looked after child. And then thinking about additional genetics appointments, say when a child has gone from being a looked after child to being adopted, is that a point when another genetics appointment might be helpful for the adoptive parents who have perhaps missed out on the information that was made available to the foster parents, for example. We're going to think about pathways, sort of sharing information, how people make referrals, all of that sort of practical information that's really helpful. Then again, sort of thinking about providing that sort of accurate information about, for example, including information about genetic testing that might be appropriate in the future or that a person might want to consider in their later life letter, which is provided when they sort of might leave the looked after system. So that's just a summary of what we could improve. And that's me. So any questions?
Oh, I probably don't need that. Most people don't. Yeah, it's fascinating. Real ethical problems you're going to have though, aren't you? Because you may have family members who are actually still alive, but separated. And how do you test one and the implications for the other? And,
Absolutely, yeah. Yeah, so it's something we already think about fairly regularly in genetic services, but you're absolutely right. It can be really, really difficult to think about what's best for the child or the young adult or the adult who's been in the looked after child system, and what implications that might have for biological family. Yes, you're absolutely right.
There are also implications for the adoption service in terms of their knowledge of a child having a genetic.
Yeah, and again, that's something we get fairly regular contact with social services around. Is about testing, for example, for a genetic condition that we might not normally test for in childhood, because there are sort of thoughts that it might have implications for whether that child might be adopted. And yes, yeah, it's a really sort of complex and difficult area. But that's why I think setting up those connections might really work, hopefully, to go through those questions and think about what's going to be best for the child at the centre of it. Yeah.